Hello, everybody. It is my great pleasure to introduce our next speaker, Professor Nicole Rust from the University of Pennsylvania, where she's also the director of the Visual and Memory Lab. Dr. Rust's research combines behavioral, neural, and computational approaches to understand the brain's remarkable ability to remember the things we've seen, focusing on where and how visual memories are stored. As a complement to this foundational research, she also works to develop new therapies to treat memory dysfunction. Dr. Rust's work has been recognized by many awards, including the Troland Research Award from the National Academy of Sciences, the McKnight Scholar Award, a Career Award from the National Science Foundation, the Alfred Sloan Fellowship, and the Charles Ludwig Distinguished Teaching Award. I've also recently learned from her website that she's currently writing a book. And that book is about brain research, and she hopes in this book that she would inspire multidisciplinary and community-based conversation. So let's all welcome Dr. Nicole Rust.
Thank you, Kalanit. Thank you so much to the organizers. It's a great symposium. I'm really part of this. Yeah, real pleasure. So I just want to begin. I just want to begin with the observation that it is such an amazing time to be a brain researcher right now. There are so many things, and they're happening so quickly. There's so much opportunity here. It's like a fire hose of opportunity. And I think at times like these, it's really important that we pause and we stop and we ask ourselves, what is it that we're trying to know? So what are we trying to do here when it comes to memory? What do we want to prioritize understanding? And what is our plan in order to obtain that knowledge? And I would say that, for a long time, the approach to memory has been very focused at what's happening. So our approach to memory has been focused very, very much at the molecular and cellular level for a good reason. So we focused a lot on neurotransmitters and synapses. We focused on how molecules combine to form neurons, how neurons combine to form circuits, and how circuits combine to determine memory function and dysfunction. One classic example of this is Alzheimer's disease, where the predominant hypothesis is that there's a molecule, beta amyloid, which is a toxic substance. It triggers a degenerative cascade in memory neurons and circuits, and that's what leads to memory function, memory dysfunction in Alzheimer's patients. So I've combed through the abstracts that are currently supported by NIH and NSF to try to get a sense of how are different memory researchers approaching what they're doing. So just randomly taking a subset of those abstracts and trying to wrap my head around what are the approaches. And my estimate is that about 70% of memory research that's happening right now is well described in this way. So we're still very much targeting what's happening at the level of molecules and neurons. But you might notice that I've left a gap here on this slide. That's very intentional. This gap is a gap that Matteo Carandini coined a bridge too far. So it's this very nice paper he wrote about a decade ago. If you have not read it, I encourage you all to read it. It's a joy to read. What lies in the gap is embodied in this classic quote by David Marr. Trying to understand brain function by studying only neurons is like trying to understand bird flight by studying only feathers. It just cannot be done. Okay? We can't just understand neurons and try to understand brain function. What lives in the gap is often called systems and computational neuroscience. It's a difference between what the brain is computing in order to support memory function versus how the brain is executing those computations in its biological machinery. And I'd like to argue that we need to fill this gap for memory. It's a really important gap to fill. We need to fill that gap even when we believe that our interventions, as is the case for Alzheimer's disease, are going to happen back at the molecular level. The only good way we're going to arrive at those interventions is if we fill this gap. Okay, so this is one of the reasons I'm so interested in this gap. It's also the case that we have all of these emerging therapies for treating memory dysfunction, none of which exist yet clinically, but all under development that go under this name neuromodulation. So examples include deep brain stimulation, vagus nerve stimulation, brain-computer interfaces, they're all targeting this level brain activity directly. So we're going to have to understand this level in order to develop those therapies as well. Okay. So today I want to focus on this level using the work from my own group as an example of how we can go about understanding memory by targeting what's happening here. And I'm going to focus on a particular type of memory. It's often called familiarity memory or recognition memory. It's what you tap into if we show you an image like this and we ask you, have you seen this image before? Yes or no? And one of the things that motivates us to study this type of memory is its translational clinical potential, right? That we think this is a very tractable form of memory, that we have some hope of figuring out, maybe on a short time scale, and we can leverage that knowledge about basic memory function in order to develop new treatments for memory dysfunction. We're also really interested in this type of memory because it turns out that humans are just really, really good at it, and we have no idea really why that's the case. So we're really good at remembering whether we've seen images before. And documentation of just how good we are at this goes back to this classic paper, which is aptly titled Learning 10,000 Pictures, published by Lionel Standing in 1973. But this also continues to be studied in contemporary labs like Ode Oliva's group at MIT. So these experiments, they're pretty straightforward. So they begin with an exposure phase where you have people look at photographs. They'll look at them each only once, each for a few seconds. The key in these experiments is they need to look at thousands and thousands of images over the course of hours. At the end of this exposure phase, you then give them a memory test. You place two images side by side, one they've seen, one they haven't, and you ask them, which one did you see? And the consistent observation across many studies is that people are really good at this. So for example, in this Brady study, subjects viewed 2,500 images over the course of 5 hours. They did get to take breaks, but over the course of 5 hours. Then they're given this memory test, and they're over 90% correct at remembering which of those two images were embedded in that stack of 2,500 photographs. Even more remarkable, I think, is that subjects continue to be really, really good at this when the two images placed side by side are very visually similar to one another. So note these two images are both bread. It's in a bread box. In one case, the bread's a little bit more in the front. In the other case, it's a little bit more in the back. Subjects weren't quite as good, but they were nearly as good, 87% correct, at picking out which of those two images was embedded in that stack of 2,500 images they'd seen each only once and each for a few seconds. So what this tells us is we're storing these memories in our brains, not just the objects we've seen, I saw bread, but a lot of rich visual detail, the context, the configurations that we're seeing the objects in. Okay. So just to summarize this type of memory, it's an example of single trial learning. So we remember images after seeing them each only once, each for a few seconds. It happens with vast capacity, so we can remember thousands and thousands of things. And it happens with remarkable visual specificity. We really can remember whether we saw the bread in the front versus the back of the bread box. So the big question that we're interested in answering is, how do our brains manage to support this type of learning? So right now, my lab is all in on trying to address this question in various ways. We study human behavior. We also study brain activity, not in the brains of humans, but in rhesus monkeys. And the reason we work with rhesus monkeys is because they're the only animal model that to date has been demonstrated to be capable of anything like this. Okay? By that I mean seeing large number of images, each only once, and reporting whether or not they've seen them before. No other animal model has been capable of being able to report that. And I can tell you, it's despite our extensive efforts in trying to make that happen. It's really only working in rhesus monkeys. And in the brains of rhesus monkeys, we can record with that exquisite spatial and temporal resolution from large populations of neurons that we think is absolutely essential for answering this question, how do our brains manage to support this type of memory? And so today in my talk, I'm going to speak to the question, how does the brain transform seeing an image to remembering an image? And I'm going to break this down conceptually as happening across 3 different stages. If I show you an image and ask you, have you seen this before, yes or no, this begins with seeing. This is followed by memory storage. And then your brain has to convert that information about memory into a behavioral report. That's what I'm going to call remembering. You might think of it as a decision process, right? And so we're going to ask the question, how do these different stages unfold in the brain? Now, in the brain, we have a pretty good idea of where we should look, or we think we have a good idea. So in terms of seeing, that begins in visual cortex. So signals travel from the eye and then up into visual cortex, V1, where they propagate through the ventral visual pathway up into inferotemporal cortex, IT. After signals leave IT, they go into the medial temporal lobe, including perirontal cortex, which receives its primary input from IT, and then up through the medial temporal lobe into the hippocampus. And today I want to speak to the question, what is the role of the medial temporal lobe in shaping this type of memory? So for those of you in the know, this question has a very long and controversial history toward it. Today, I want to address this question by specifically targeting the problem, does the medial temporal lobe shape our ability to remember whether we saw the bread in the front versus the back of the bread box, the remarkable visual fidelity of visual memory? And it is hypothesized that yes, indeed, it does. So the current hypothesis that's out there is that the hippocampus is contributing to this behavior through a computation that's called pattern separation. I'll describe exactly what that is in just a moment. But for now, I want to highlight that this is a computation that is thought to happen downstream of visual cortex and thought to contribute to reformatting the visual information in a way that prepares it for memory storage, that makes these visual representations, these visual memory representations more robust. So in part one of my talk, I will describe our tests of this hypothesis. And I'll present some evidence that when we do these experiments, it really looks like the hippocampus is inheriting these high fidelity visual representations. It's not actually participating in the computation of them. This really calls this computation into question when it comes to this particular behavior, right? Visual familiarity memory. But in part two of my talk, I'll present evidence that the medial temporal lobe does seem to be doing something when it comes to shaping this type of memory, and that is it's contributing to disambiguating memory signals, memory information, from other types of information, okay, like visual information, stuff that you don't care about if what you're trying to do is a memory task. And these computations are things we can think of as happening downstream from the locus of memory storage, involved in the process of remembering itself, but not memory storage per se. Okay, so that's my agenda today. Before I dive into all of the details, I just want to emphasize why we care. So we care because we're really interested in using platforms like these as an opportunity to develop new neuromodulation therapies. And when you want to do that, you really care where in these three conceptual stages you are. For example, if you want to develop a neuromodulation therapy that maybe targets the hippocampus and uses a behavior like this, you really want to know, are you before the locus of memory storage? Are you at the locus of memory storage? Are you after the locus of memory storage? That's going to determine whether or not these therapies work or they don't, right? So that's why we care so deeply about these questions. Okay, so part one, testing the hypothesis that the hippocampus contributes to this behavior via pattern separation. So again, let me just highlight. The question is, what in our brain shapes the high fidelity of this type of memory? And let me just describe to you the hypothesis of how this works. So it all begins with seeing, where we know that if you look at an object, like a coffee cup, that's going to activate your eye. It's going to activate this wave of activity across your brain. And in high levels of visual cortex, places like inferotemporal cortex, IT, I like to call what we see there as a pattern of spikes representation. This simply refers to the fact that the cup will activate some neurons more than others, and you will get a pattern of spikes across a population. You change the image that you're looking at, you'll change the pattern because they'll activate different neurons. And consistent with a lot of what's happening in systems and computational neuroscience right now, we like to interpret or decipher these patterns of activity in these high-dimensional geometric spaces. So the dimensionality of these spaces can be up to the number of neurons that are included in the population. So in the case of IT, up to 16 million dimensions. But we can make inferences about what's happening in these spaces by just looking at a two-dimensional space, say the vigor of the response of unit 1 plotted against the vigor of the response of unit 2. And a particular pattern of activity will correspond to a vector that's pointed in a particular direction in this space. Change the pattern, you change the direction that the vector points. And this really is how we think IT is working, right? The brain's just looking down on IT and seeing what direction are these vectors pointing and inferring the content of what's out there in the world. And it's the case that in IT, when you have perceptually more similar images, you have close vectors that are closer together in their directions. Okay. So that's seeing. And now the question is, well, what happens next in memory storage? And so we begin by acknowledging that memory storage is a noisy information degrading process. That's important. And the hypothesis is that the amount of overlap in high-level visual cortex is too much. There's too much overlap to support the remarkable visual fidelity of visual memory. If you add noise to those two vectors, they're just going to collapse on top of one another. So what needs to happen next is a computation like pattern separation, which is proposed to happen in the dentate gyrus of the hippocampus, which is, there's a cost here. You're going to lose information when you do it, but the result is that you get vectors that are farther apart, they're more separated, and you do this before the locus of memory storage. So now if you add noise, these vectors don't collapse on top of one another. That's the idea here. And there's tremendous evidence in support of this hypothesis. It's quite convincing. This includes, for example, some early evidence from Craig Stark's lab, which set up a really nice behavioral paradigm to measure this. So in this behavioral paradigm, people are going to see images, and then either what will be repeated is either the same image or a visually similar lure image. And these lures vary in their degree of similarity, which is organized into bins according to the amount that they'll confuse memory. It's called mnemonic similarity. So this picnic basket here will be less confusing. So let me just be clear. What subjects see first are the top row items, and then what is repeated are the bottom row items. And subjects are less likely to confuse the picnic basket when what they saw was the other picnic basket, and they're more likely to confuse the rubber duck when what they saw was the first rubber duck. Okay. And the observation is that patients with hippocampal damage will have actually normal memory if you show an image and you repeat the exact same thing, right? So overall, this type of memory doesn't seem to rely heavily on the hippocampus, but patients will be impaired for these visually similar lures. So they'll actually overgeneralize their memories, such that if they see the picnic basket, they tend to say, yes, I saw that before, at a higher rate than normal healthy controls. So this suggests that the hippocampus is in fact playing some sort of role in shaping the visual fidelity of visual memory. There are also some really nice fMRI studies that reveal higher fidelity memory representations in the hippocampus as compared to earlier structures. All of this made us very optimistic that this is a task that really taps into hippocampal function and one that we can use to build our neuromodulation therapies upon. But before we did that, we wanted to go in and replicate these results in monkeys and also develop a modification of the paradigm that allowed us to tap into the amount that sharpening was happening downstream of IT. We were able to do this because we now have all of these really great models of how IT will reflect images. These are the deep artificial neural networks. They're not perfect. They're pretty good predictions of how images will evoke responses in IT. And we can build on those to create these benchmarks of what we would expect IT to do. And then we can measure how much sharpening is happening downstream of those IT representations. So the work I'm about to talk about is the work of a talented graduate student, Barnes Januzzi, and he created image sets that had novel images, as shown on the top, and then what would be repeated in the memory experiment are either the same images or visually similar lures that vary in their degree of dissimilarity, benchmark to the responses of IT cortex. Okay, so these are, for those of you in the know, this is representational similarity of a deep artificial neural network. So we've tested humans in these experiments. We've also tested monkeys. This is the monkey version of the paradigm here. It begins with a monkey looking at an image on each trial, for 500 milliseconds, and then they make a behavioral report, whether that's novel or familiar, with an eye movement response to one of two targets. So for this monkey, an upward saccade means novel. Monkeys will see the images first as novel, and then repeated later in the sequence as familiar, either a exact repeat or a visually similar lure. And novel and familiar images are continuously intermingled throughout the session. So then we get a behavioral plot where we can plot the percent of times the monkey will say that an image is repeated as a function of how visually dissimilar it is from what he actually saw. Hits are measured based on exact repeats. So these are images that are repeated with zero visual dissimilarity. False alarms are based on the novel images. So these are images that are visually distinct from anything else he's seen so far. And then in between, we have lures at varying degrees of visual dissimilarity. And so those are intermediate. And importantly, in these experiments, we can benchmark relative to what we would predict would happen based on representations in IT, assuming that false remembering of lures falls linearly as a function of representational distances in IT. That may or may not be true. That's an empirical question. But it's important that we have this benchmark so we can measure how much sharpening is happening beyond the IT representations. It's also the case we're using a deep neural network to create this benchmark, but we have neural data, and we can verify that the model is doing a good job. So sharpening behavior, which would be what would be predicted from a system that implements pattern separation, would look like this downward bow here. So we measure this in humans, we measure this in monkeys. It's a very reliable and robust behavior. I'll show it to you for one monkey. Here you can see in the thin lines the behavior from an individual session. In the thick line, the average across sessions. You can see we're seeing very reliable sharpening behavior relative to this IT benchmark. Okay. And so now the big question is, well, is this behavior following from sharpening that's happening via some sort of processing in the hippocampus? It could be pattern separation, it could be something else. So to address that question, we recorded, I should say Barnes recorded, neural responses in IT and the hippocampus. And he's going to then look at the memory representations in each of those structures and look to see the degree to which they align with behavior. In order to look at the memory representations, we use the following approach. So we consider the responses to the same image when it was first presented as novel, and then it was repeated as familiar, and we compare them. And our empirical observation is that, at least in IT, what you get is an overall reduction in the vigor of the response when you repeat an image. It's an adaptation-like phenomenon known as repetition suppression. But you don't change the pattern of the response, really, in any obvious way. Okay. We've done a lot of work to demonstrate that this repetition suppression signal seems to be nicely aligned with behavior in a number of different ways, making it a good candidate for the so-called memory signal. In our geometric framework, what this amounts to is population response vectors that are longer when an image is novel, and they shrink but don't change their direction when a stimulus is repeated. Whether or not this is what happens with memory or something else, our approach will be to linearly decode these memory representations by comparing the responses of images when they're novel as compared to familiar, and then determine the degree to which those memory representations in each brain area align with behavior. So let me show you the results. So here I'm just replotting the behavioral result I've already shown you in the previous slide for one monkey. And now if we do this decoding prediction based on the hippocampus, we see a nice alignment with behavior. So what's being reflected in the hippocampus really does reflect the sharpening behavior we observe by the monkey. Surprisingly, that's also true in IT cortex as well. So in IT, we also see sharpening behavior. And I want to emphasize this plot was created using spikes that were counted late in the viewing window. So the monkeys viewed the images for 500 milliseconds, and here we're counting spikes 300 to 500 milliseconds after stimulus onset. We can also look earlier in the viewing window at 125 to 325 milliseconds. There we see IT is also really nicely aligned. And the hippocampus is doing something surprising here. It's broadening, it's not sharpening. So a parsimonious interpretation of these results would be that the hippocampus is actually inheriting these high-fidelity visual memories from computations that are happening back in IT, as opposed to actually computing them, right? There's no computation really that needs to be done after signals leave the IT. It's already been done. So it's inheriting versus computing. And I just want to emphasize that I think there's a whole lot of work to be done here to tease apart why we're seeing results that seem to be distinct from what was being observed in those human studies. I think there's a lot to be done. We're very invested in doing it. But from the big picture perspective of we're looking for a task that taps into hippocampal function, again, in order to develop this platform to develop neuromodulation therapies, this doesn't really seem to be the right task for us, right? At least under these conditions. Okay. So again, we started with this behavior. Do you see the bread in the front versus the back of the bread box? The remarkable visual fidelity of visual memory, and the hypothesis that the hippocampus has contributed to shaping this behavior through a computation known as pattern separation. We tried to test that hypothesis, and our results suggest that the hippocampus is inheriting these high-fidelity visual memories. It's actually not contributing to their computation per se. And this begs the question, well, is the hippocampus doing anything when it comes to tasks like these? Is it contributing in any way? That's why I want to share to part 2 of the talk. Well, I guess I should say, yeah, so this calls kind of this into question. So now in part 2 of the talk, I want to turn to evidence that, yes, the hippocampus does seem to be contributing something, but it's not quite what we thought. And that is the disambiguation of these memory signals from other things, and these are computations that we can think of as happening downstream from the locus of memory storage. Okay. So now I'm going to switch gears and turn to a different behavior. Instead of the fidelity of visual memory, I want to focus on image memorability. So this simply refers to the fact that in these tasks, we systematically find some images easier to remember than others. The images on this slide are configured according to their memorability. So the most memorable images up here, down to the least memorable images down here. And Ode Oliva's group has come up with really nice ways to quantify the memorability of images and to demonstrate that that actually means something. So each image gets a memorability score, which reflects loosely the fraction of subjects that remember seeing it. So there are some images out there, like the ones in this top row, that people are really good at remembering. So 85 to 90% of all people will remember them. There are other images, like this bottom row, where people mostly forget them, like that nature scene there. Only 44% of subjects will remember seeing that one. And importantly, these scores are reliable and predictive. So if you measure them for one group of human subjects, they are very predictive of how well another group of human subjects will remember images. Stated differently, we all tend to find the same images memorable and forgettable. And surprisingly, it's not just us, but we've also tested monkeys. And what monkeys find memorable and forgettable is correlated with what we find memorable and forgettable. So monkeys are actually a good model for studying this. We think this follows from just the structure of their visual system, which we already know is structure and function that's much like our own, as well as much of the structure and function of their memory systems. Okay. So again, we can ask for this signature of this behavior, what makes an image memorable and forgettable? How does this unfold across these different processing stages, seeing, memory storage, and remembering itself? And ultimately, what I want to get to here is what is the contribution of the medial temporal lobe to shaping this behavior? But in order to do that, I first have to tell you about, quickly, a handful of studies that we've been doing across the last few years in order to inform these earlier stages. So I'll just start with seeing. So I want to pick up where we left off, where IT cortex reflects the identity of what you're looking at as the angle or direction of these population response vectors. A few years ago, we asked the question, well, how is memorability reflected in IT? What differs in IT in the representation of a more memorable versus a less memorable image? And the answer is refreshingly simple. It seems to be the overall vigor of the response. So more memorable images evoke more vigorous responses, which correspond to longer population response vectors. Okay. So that seems straightforward. And then the question is, well, how does that relate to a more robust memory? Remember, in IT,
I introduced this idea that memory is also reflected in the overall vigor of the response. So novel images have more vigorous responses than familiar images. We've determined in unpublished data that those more vigorous responses that you see the first time an image is presented produce stronger memory signals. They produce stronger repetition suppression. Okay. So this seems pretty straightforward, right? More vigorous response, bigger reduction. It's kind of an adaptation-like thing. That all makes sense. The problem comes when you think through the geometry of this type of representation and what would be required to decode or infer it, you start to see confusions. And they're easiest to think about. It's true of memorability, but it's easiest to think about with contrast, and that's actually where we've done some experiments. So higher contrast images also evoke more vigorous responses in IT. But no, we don't systematically confuse contrast and memory. So we don't see a high contrast image and think it's familiar, think it's novel, or see a low contrast image and systematically think it's familiar. So the question is, what is going on there? So in some experiments we published last year, we inferred the existence of corrective computations downstream of IT that serve to isolate these memory signals. The general idea here is that you want to get rid of this nuisance modulation and extract the memories. So that leads us to the hypothesis that this is exactly what the medial temporal lobe is doing. It's performing these corrective computations. Okay. The idea here is that this is the hippocampus. So I'll quickly just wrap this up and say that this hypothesis makes a number of predictions that were tested by a talented graduate student, Simon Baum. The first is that total memory information is preserved between these two brain areas. So this is a feed-forward cascade. Should we find that there's more memory information in the hippocampus than back in IT, that would violate this prediction. Memorability modulation has been removed. That's a whole point. And that you can simply look down on the hippocampal population, count the total number of spikes, and determine whether or not an image is novel or familiar. And I'm running out of time, so I'll quickly just say each one of these predictions has been verified in our experiments. In these experiments, monkeys look at an image on every trial. Images have different memorability. We record neural responses in IT and the hippocampus. Total memory information, which we have ways to measure, as a function of population size, it grows in IT at a particular rate, which is matched in the hippocampus. There's no more memory information in the hippocampus than there is back in IT. The memory modulation has been removed. There is no memorability modulation in the hippocampus. And if we try to decipher, just by the overall vigor of the response, how memorable images are, here's an individual monkey's behavior. So this is just a plot that says this monkey chose familiar at a higher rate when images happen to be more memorable to the average human population. So those two are correlated. This is memorability behavior. The IT prediction is misaligned. It has the wrong slope. It's obviously not right. But if we decode information from the hippocampus, we see this nice alignment. Okay. So I just want to summarize the big picture here. So in the transformation from seeing to remembering images, we think it unfolds like this to describe memorability. More memorable images evoke more vigorous responses in IT. Those more vigorous responses create more repetition suppression, but this creates a representation in which vision and memory are all jumbled up together. And then corrective computations downstream from IT in the medial temporal lobe create hippocampal representations where you have an isolated memory signal. That's how we see this unfolding. My final point, I just want to emphasize that these corrective computations, they amount to something like a decorrelation. So when an image is high contrast, all the neurons are going to respond a little bit more. And when it's low contrast, they're all going to respond a little bit less. That's what I mean by decorrelation. Those are stimulus-driven correlations. That's what this computation is removing. It's consistent, I think, with some other models that the hippocampus is performing some type of compression. And it's a computation that could be easily confused with pattern separation, but I think it's a distinct computation, and hopefully you have a sense of how those two things are different now. Okay, so I'll just end there. I want to thank everyone who contributed to this work. At least one of the studies on the slide, each of these players were involved, and I just want to also thank the funding agencies that support this work. So thank you very much.
Thank you, Nicole, for such a wonderful and inspiring talk. And now let's take some questions. You have a question here? Yeah.
I have, okay, after.
Yeah, go for it.
Hi, I'm Laura Williams from UCSF. Thank you for such a beautiful talk.
Thank you.
It's very. So I wondered about the visual dissimilarity metric that you were showing on the x-axis. You said that you derived that from neural networks. I wonder if you could give a few more details. Seems that there's a number of ways that you could derive that, and that's quite important.
Yeah. So this particular measure is representational similarity measured as the correlation between two population response vectors. Yeah. I also want to add, if we can replicate this behavioral result if we replace the actual IT responses we recorded with the model predictions. It also replicates. Yeah, it doesn't change the story. So we verified it in that sense. Yeah.
Thank you.
Okay. Jay?
Hey, Jay.
This is really fascinating work, and I'm really impressed with your clarity of your pattern separation results in IT. But I just want to probe your thinking about the locus of storage of the memory. So from my point of view, what's stored is not the pattern, but the changes in synaptic strengths among neurons. And the fact that the pattern is already separated in IT doesn't tell me that the synaptic changes that involve the storage are happening in IT. They could still be happening in the hippocampus. So my story would be the hippocampus is doing this beautiful job of taking out the the overall intensity signal as a very, very helpful step towards having the representation, which is then just the right one for the synaptic changes that would then still be occurring in the hippocampus.
Yeah.
And I just wonder if that's consistent with your way of thinking, or how you...
Yeah. No, thanks for the question. So I just want to emphasize going into this that we've tested this under a certain time course, right? Which is the time course of memories that are stored for minutes, but not hours or days. And in the conditions we've studied it, the empirical finding is that repetition suppression is the signal that seems to be really nicely aligned with behavior, and there really are no other candidates on the table that are. So then the biophysical question is, what creates that phenomenon known as repetition suppression? And there, I think there isn't a single factor. I think there are multiple factors. So this repetition suppression is actually observed as early as the retina. It happens a very little amount. It happens at a very short time scale, but it's there, right? And if you go through the ventral visual pathway, you get a little bit more of it at a little bit longer time scales at each stage of the cascade. So I think in IT, we're seeing those cumulative effects within IT and up to IT. There's also some nice models to suggest that there's probably some recurrent computation happening in IT itself. But what you're asking about, is there any component of this that is fed back from the hippocampus? And I don't definitively know the answer to that using these experiments. What I can observe is there's nothing in the hippocampus that aligns nicely with behavior first. And in fact, what we're seeing is the opposite. We're seeing, the thing that's aligning with behavior is showing itself back in IT before it ever shows itself in the hippocampus. So I would love to sit down and talk with you in more detail about, because I think what you're saying is, well, I think pattern separation happens, and then there's some memory thing, and then there's a feedback. And so we could talk about more elaborate models and whether or not we have any power in our data to discern between those models. But what I've presented today is, I think, a parsimonious view of what seems the low-hanging fruit that seems to be going on.
Okay, so maybe Elena has a question? Yeah.
Hi, wonderful talk. My name is Alina. I work with Colonnade Grille Spectre as a postdoc. You alluded a little bit to this in your answer to Jay's question. I was wondering, so seeing itself is an interpretation already. It's not just a camera, just feeding in images.
Yeah.
So I was wondering, how do you think about not feed forward information, but the feedback, the fact that it's more memorable, a more memorable image itself? How do you think about that feedback? Or like,
Yeah, I mean, I think there's clear evidence that it's there, right? Like Koji Carr has this beautiful evidence from when he was working with Jim DiCarlo that if you inactivate prefrontal cortex, you disturb behavior, you disturb neural responses in IT, right? So I think a lot of us are starting to think about what's happening in the ventral visual pathways. There's this feed-forward wave that is kind of reshaped via feedback, and you've hit on the conditions I think are most important, and that is under conditions of ambiguity, right? Where you're really trying to figure out what you're looking at. So clearly, the brain is full of complex recurrent loops. I think we're only just starting to scratch the surface and trying to figure out how to pick apart causality given a complex recurrent system. And so really excited about the field moving forward in terms of that. How much of that is happening here? I think, yeah, it's really an empirical question, yeah. And there's, the fact that things are happening in IT before they're happening in the hippocampus, right, I think puts a strong constraint on some of the data, yeah.
Okay, Tony.
Thanks, Nicole. That was really beautiful talk.
Oh, thank you, Tony.
So I'd like to go back to a slide. This was the first one where you had lures.
Okay.
And it seemed to me that the breadbasket lure is two different objects.
Yeah.
But the duck lure was a rotation of the same object. And the glasses were a rotation of the same object.
So not our images, but the...
Yeah. And it just
Yeah,
triggered this
yeah.
question. So you can create image dissimilarity in two ways, one
Yeah.
by swapping the object and the
Yeah.
other by rotating it.
Yeah.
And that would seem to have big implications for whether you should do pattern separation or not.
Yeah. Yeah.
Maybe you could speak to that. Because it's the invariant object recognition is the task, really
Yeah.
recognizing images as being different or objects being different.
Yeah. You're speaking to experiments that Simon, who did the memorability work, is developing right now. So yeah, so I can ask you, have you seen this image before, where, like you said, two images that have the same object but are distinct? You want to say, no, I haven't seen that one. I saw a different one. But you can also ask, have you seen this object before? And in that case, all of these computations are the opposite of what you want to do, right? You want to generalize memory. You don't want to shape it to be sharper. And that might depend on, is the dissimilarity created by the background or is the dissimilarity created by the object? And so so Simon is just starting to develop that experiment and that line of thinking and trying to tease those different things apart. But I think the big question there is a fascinating one. If everything is processed by these same circuits, then the things that are making things sharper are working against you for doing a different task. And so something has to give there. And so where is the flexibility in the system? Is it something like attention? Is it something like just different modules that are processing different things? That's exactly the question I think that needs to be asked next. Yeah, thanks.
I think there's another question up here. We have the mic, please, sorry. Okay, we'll do the question here and there. We have two more questions, thank you.
Three more, actually.
Three more, okay. Three more questions.
Great talk. I was kind of curious about where the electrodes were placed in the data, given that so much of the pattern separation literature is kind of focused on the dentate gyrus and, and CA1, even different parts of CA1, distal versus proximal, are doing, different processes.
Yeah. In these experiments, we're in CA1. Yeah, we're currently exploring CA3 to see if the answer is different there. But the dentate is just so hard. I would say some combination of the dentate and CA1. It's hard. It's really hard to differentiate them. Yeah.
Do you have enough span for CA1 to see if you look at electrodes in distal versus proximal CA1, given that more distal CA1, which is actually don't know what it's like in the primate brain.
Yeah, in the primate, it's a good question. We're still just digging into the data, but it's a really good question, and we should ask ourselves. We always try to spread our recordings out as much as we possibly can, but do we have any power in the data in order to do subsets of the analyses? That's a great question. Yeah.
Okay. So there are two more questions in the back.
Thank you. Thank you for presenting today. You talked a lot about sharpening and broadening happening in the hippocampus. Could you better define what sharpening and broadening is?
So in our experiments, we set it up directly to test this hypothesis. here that it is a nonlinear transformation that is happening downstream of the IT representations. And it's a nonlinear transformation that enhances the fidelity of visual memory ultimately. So that would be my definition. And we created that benchmark based on the IT representations themselves.
There's one last question. Oh, you have a question. Sorry, go ahead.
What non-linear fitting or regression did you use?
We measured, so we had a benchmark prediction of what would happen in IT cortex. And then we measured sharpening relative to that benchmark. So I'm not showing you fits. I'm just showing you that every single session we're getting a downward bend relative to that line. So I think hopefully that's pretty convincing. We could fit something to it, and I could try to convince you statistically that way. But I think the answer is pretty clear from, yeah.
Thank you for a nice talk. I actually had a question about this figure. You showed it also for tens of milliseconds differences. And you showed the various brain areas. I'm just wondering how that looks if you were to maybe split the beginning of a session versus the end of a session. Do you see the kind of this curve changing differently for hippocampus? Maybe like you're, I don't know, getting worse at separability and maybe hippocampus than you are in IT?
Yeah, there's some kind of a loading effect.
Yeah,
it's a good question. I don't know the answer to that. It's a great question, and we could try to do that analysis, but we haven't tried anything like that so far.
Thank you.
Thank you, everybody, and thank you, Nicole, for your wonderful talk.
Thank you.